So we're very pleased to have Professor Pieter Roelfsema with us today in our Bar-Ilan University Vision Science Seminar. Peter is an MD and has a PhD that he has received from the University of Amsterdam after doing his research at the Max Planck Institute for Brain Research in Frankfurt in Professor Wolfsinger's lab, where he already started to investigate the functional role of neuronal synchronization in the visual cortex. He did two simultaneous postdocs, one at Professor Wolfsinger's lab and another at the University of Amsterdam in the group of Professor Spekreetje. I'm not sure I'm pronouncing this correctly. He took part in organizing many conferences and symposia, and among his executive roles, he's the head of the Vision and Cognition Department in the Netherlands Institute for Neurosciences and the director of the Netherlands Institute for Neuroscience. He's also the chairman of the Neurotech NL and a co-founder of the Phosphenix, a company that aims to build visual cortical prosthesis for the blind. He collaborates with researchers across the world. He published more than 100 highly cited papers in top leading journals as Nature, Science, PNAS, Cell, Neuron, Trends in Neuroscience, and many others, and has more than 16,000 citations. As I've already mentioned, he's a professor at the Netherlands Institute for Neuroscience in Amsterdam, a professor at the Free University of Amsterdam, and also a professor at the AMC in Amsterdam. His research tries to understand how neurons in different brain areas work together during thinking, how networks of neurons work together to solve cognitive tasks, and how they configure themselves during learning. He studies visual perception, plasticity, and memory in the visual system of experimental animals, humans, and with neural networks. He also develops neurotechnological solutions to create new high bandwidth and high computer and brain-computer interfaces, and combines new technologies, new technological possibilities with knowledge about the visual system to create a visual prosthesis for the blind to restore a rudimentary form of sight. So without further ado, I want to welcome you, Peter, to our seminar, and thanks for joining us.
Thanks, Sharon, for a very nice introduction. So I'll share my screen so we can get going. Let's go to slide 1. Okay, let me know if you can see my screen, if it's okay.
Yeah, definitely.
Yeah, so today I would like to cover several topics that are intimately related to each other. So the first question I would like to ask is, how does a weak stimulus enter into conscious awareness? And I hope to convince you that the critical step is getting access to a working memory. And I would like to discuss how this then works in the brain. And then I will go from detection of very simple stimuli to more complex stimuli and talk about perceptual organization, perceptual grouping. And in the last 10 minutes or so, I'm going to talk about using this knowledge to create an interface with the brain of blind people trying to restore a rudimentary form of vision. Now feel free to ask questions at any time. I won't be able to see the chat, but maybe Sharon, you can see it, and feel free to interrupt me whenever appropriate.
Yeah, I will, sure.
So before that, I'll start with a brief introduction, talking about feed-forward and feedback connections and the type of processing that you can expect from them. This will be relatively brief because I think everybody or most of you will know this. So this is quite a famous slide by Velleman and Van Essen. Already 30 years ago, they published this famous paper. This is the brain of the macaque monkey, and they gave colors to all these different areas of the macaque monkey involved in vision. And from the same paper, there is this interconnection scheme where they arranged all these areas into a hierarchy where information starts at the retina, propagates to V1, and then to higher visual areas through a network of feed-forward connections. And we know that these connections allow low-level areas to code for simple features, such as the orientation of a light bar. Neurons in areas at intermediate levels code for feature constellations of intermediate complexity. And if you ascend the hierarchy even further, you'll find neurons that code for specific categories. Now, there are not only feed-forward connections, there are also feedback connections, allowing for recirculation of information. And in the second part of my talk, I would like to discuss how they contribute to perceptual organization, basically our ability to group together all those image elements that belong to the same perceptual object. Okay, that was my introduction. That was easy. Okay, so now we're going to talk about visual awareness, and I'm going to use a very simple method that have been used by others. And that is, if you present a very weak stimulus, sometimes it will make it into your consciousness and sometimes it will not. And that's a very nice method, because then you can compare the very same stimuli and see what is the difference between those that made it and those that did not. So now I can't see every one of you, but I'm going to entertain you with a very simple task. I'm going to ask you to look at the fixation point, and about here there will be a very simple stimulus. If you see the stimulus, you might want to raise your hand. If you don't, well, don't raise your hand. It's very simple. Okay, so are you ready for this? 1, 2, 3. Yeah, so it all works. That's good. I'm going to do it again, make it much more difficult. So this is for the careful viewers. 1, 2, 3. Okay, nobody saw it. Once more, 1, 2, 3. Yeah, it was barely visible. 1, 2, 3. Maybe somebody saw it. Now I will be honest with you, there was nothing on the second and the fourth presentation. There only was a stimulus on the first and the third presentation. You may have suspected this. And this is my introduction to a theory that is very famous, developed by Green and Sweats many years ago. It's known as the signal detection theory. And the signal detection theory holds that if you present a weak stimulus, then across trials there will be an internal distribution of signal strength that is described by this green distribution. So if you then present a stimulus, you will sample from this distribution on a particular trial. And now, according to the signal detection theory, there's also a threshold. And if this internal representation is stronger or higher than the threshold, you're going to say yes, it will be called a hit. But there's of course noise in the system. So sometimes it may happen that with the very same stimulus, you stay below the threshold, you say no, called a miss. And on those trials without the stimulus, you're going to sample from another distribution. That's the black distribution, shifted to the left. So on average, signal strength is of course weaker if there's no stimulus. And typically you will stay below the threshold, and that will be a correct rejection. But very occasionally, again, because of the noise in the system, you cross the threshold, you say yes, even if there was no stimulus, and that's called a false alarm. Everybody knows this. It's also interesting to consider what happens if you make the average stimulus a bit stronger. If you then use the same threshold, you're still going to have quite a number of false alarms. So you might want to raise the threshold. You will get much fewer false alarms. And you'll see that you now have only a very small increase in the number of misses. That's basically the green distribution to the left of the threshold. But if you would use this higher thresholds for stimuli that are weak, you're going to have quite a number of misses. Okay, so that is the thing that this signal detection theory described quite well. It's this trade-off between where you put the thresholds and the strength of the stimulus. Now, in previous work, it has also been demonstrated that you can shift the thresholds by making false alarms very expensive. And if you are going to make hits very rewarding, so if hits are very rewarding, it might be a good idea to lower the thresholds. But if false alarms are going to be severely punished, you want to make the threshold higher. So you can play all these tricks. And the signal detection theory really gives a very nice account how we then respond to those changes. Now, what is not very well known is what is this internal representation of signal strength? Where is it in the brain? So that's the question we wanted to ask. What causes the variability? What causes basically the width of this distribution? And what is this threshold? Okay, so these are the questions we wanted to address. Now, we started to work on this and we found out that what we were doing was quite nicely matched to a famous theory on consciousness. It's called the global neural workspace theory. And it proposes the following, that if you have a weak stimulus, of course, it starts in the LGN, goes to V1, then to higher areas. And Standahane proposed that if it reaches the higher areas at a sufficient strength, there is a process that he called ignition, and that he proposed to rely on recurrent interactions between these higher areas, such that if you then take the stimulus away, there is still a trace as firing rates in these higher areas. Okay, so that's where I propose that it becomes part of your working memory. And if you present a weak stimulus, then this stimulus may sometimes not cause ignition, and that would cause a miss, and sometimes it will give rise to this self-sustained activity level that's working memory, and that would then be a hit. So that is kind of the thing that turned out to describe our results quite well. So here's the task that we did. So on every trial, the monkey was looking at a fixation dot in the center of the screen. And then on 50% of the trials, we presented a stimulus, just as before, a gray dot. And then there was a short delay, and the monkey was supposed to make an eye movement to the previous location of that stimulus to report that he had seen it. But there was always this other small dot on the screen that the monkey would make an eye movement to report that he didn't see a stimulus. So on the other 50% of trials, there was no stimulus, and the monkey was supposed to go to this so-called reject dot. Okay, so it's a fairly simple task. Monkeys learn this very fast. And basically, what we then did is have the monkeys on the staircase, where we started with stimuli of a relatively high contrast. And then at some point, the contrast was lowered. And when they dropped below 80%, we said now the stimulus became of intermediate difficulty. If it was, the accuracy was above 80%, we said this is an easy stimulus. And it even dropped below 40%, we said now it's a really difficult stimulus. Okay, so that's what that's the tasks, quite simple. And then we recorded from V1, V4, and dorsolateral prefrontal cortex, which is an area of frontal cortex involved also in the generation of eye movements. So that's a caveat here. We're not going to dissociate conscious awareness from the plan to make an eye movement. And when we started to do the recordings during this task, we were in for a surprise. So what you see here is the average response of neurons in primary visual cortex, area V1. So this is the average response for the difficult stimuli, for the intermediate stimuli, and for the easy stimuli. You see, of course, that the activity goes up as you increase the contrast. No surprises there. But we then compared the stimuli that the monkey reported to those that he missed. And those that he reported are in green, and those that he missed, the activity is shown in red. And we also made sure that in this comparison, the stimulus strengths are perfectly matched. So this difference is not caused by a difference in stimulus contrast. It's really caused by, well, predicting whether the stimulus is going to make it into consciousness or not. So we were surprised to find actually that the stimuli that the monkey misses give rise to a weaker response in primary visual cortex, as if on some trials information gets lost on the route from the retina to primary visual cortex. Now, what is also noteworthy is that those stimuli that were missed but were easy give rise to more V1 activity than those stimuli that were seen but were difficult. So that implies that having a certain activity level in V1 is no guarantee that the stimulus will make it into consciousness. So the information can still be lost downstream from primary visual cortex.
Peter, can I ask a question?
Yes.
I wanted to ask, I mean, I know that the activity is probably relative to baseline, but still I want to ask if maybe perhaps it's not, were there any phase shifts between the baseline activity, between the ones, I mean, could you actually determine if there was higher baseline activity for the ones that were seen as compared to the ones that were missed?
Yes, so we looked at that, and so we looked at baseline activity. We looked also at different oscillatory signatures, power in the different frequency bands, and they do have predictive power. So, but it's quite a complicated profile. So for today, I wanted to skip that, but it's actually in the paper. You can do some, you have some predictive value if you if you look at baseline activity.
Okay, thanks.
Now, if you go to area V4, nothing much changes. So maybe here, even the difference between seen and missed is even larger in V4 than it was in V1. But also in V4, you see that the easy miss trials elicit at least as much activity as the difficult hit trials. So also a certain activity level in V4 is no guarantee that the stimulus will penetrate consciousness, and it may still get lost downstream from area V4. Now then we also recorded from dorsolateral prefrontal cortex. So this is also probably where the eye movement is being planned and generated. And there the situation was categorically different. So here all the stimuli that were going to be reported gave rise to much more activity than those stimuli that were not going to be reported. And you see that on the missed trials, there's an increase in activity for stronger stimuli, but apparently even the easy stimuli that didn't make it into consciousness do not reach a certain minimum level. So here you could basically put a threshold. If the activity in this brain region crosses the threshold, there will probably be an eye movement plan, and the monkey will report it. And I think it's going to be interesting to also look at other brain areas, whether they have this sort of dichotomous response. We also looked at the trials where there was no stimulus, and the trials with correct rejections are shown in black. So then activity was uniformly low. And we also looked at the activity on trials with false alarms. So there the monkey reported the stimulus that was not there. Now in dorsolateral prefrontal cortex, we saw that the activity gradually ramped up almost to the same level as the activity on the scene trials. So here the animal is of course having an eye movement plan. He reports something that is not there. Now this gradual ramping up of activity, we believe might reflect, we cannot prove it, but it might reflect spontaneous ignitions that happen at different time points. Now, if that happens, there's actually, if there's no stimulus, there's no natural time zero. So if these spontaneous ignitions give rise to a ramp, ramping up like this, but if this ramp happens at different time points, and if you then average across trials, you get this gradual ramp. Okay, so that would be an explanation.
Peter, there are two questions in the chat. Do you want me to read them? Do you want to have a look, or do you want to answer them later?
Yeah, it's fine. We can answer them now.
Okay, so the first one is, what role did attention or inattention play in the animal missing the stimulus? How did you control for attention? This is the first one.
Yes, so I did not. So it could be that there are lapses of attention, and that would be then an explanation why the stimulus fails to cause this extra activity, and that gives rise to an eye movement plan. That it would be one part of the explanation.
Okay, and the second one is, what about the phase relative to a certain brain oscillation? There's a lot of literature showing that the phase relative to theta or alpha may influence detection threshold.
Yeah, so we looked at that, but only in a spontaneous activity. So there are some mild effects there. I mean, it's not as if the oscillations determine everything. So people tend to overestimate the role of these oscillations, is my impression. I see Rafi saying something. Rafi, you're muted.
About the correct rejection, so there was no stimulus, yes?
Yes, there was no stimulus in the receptive field.
So assuming that there is a timing, like you said, jittering timing, would you say that the activation in prefrontal cortex without the stimulus is actually in magnitude similar to the scene response?
Yes.
It just might be jittered because you don't have a...
Yeah, I think so. You see it here at the very end. It basically reaches the same level.
Exactly. So basically, we can say that the prefrontal cortex is really reflecting the movement, not the perception.
Yeah, that's what I said, right? So that's a caveat here. And it will be interesting to record from other brain areas to see whether this is eye movement response dependent. So there are all kinds of interesting variations that you can do with this experiment to see what's really going on regarding phenomenal awareness. Yeah. So when I showed these data to Stan de Hane, who had been making models of ignition, he got very excited. His models used to be quite complicated. And he said, you know what I'm going to do? I'm going to make the simplest model that can account for your data. And that's what he did. So he made a model where the brain is composed of 5 neurons, 5 brain cells only. These neurons are stochastic, and so activity starts in the LGN, then it goes to V1. There is also a self-connection here, and there's a feed-forward connection to V4. There's also a little bit of feedback then from V4 to V1. And then to parietal and frontal cortex, so in total 5 neurons. And he made the connections between the parietal and frontal neurons relatively strong, and also the self-connections relatively strong, so that if there was a minimum level in parietal and frontal cortex, the activity could self-sustain. Okay? So, and if that happens, we can call it ignition, then the activity will become self-sustained. And if you take away the stimulus, there's basically a memory trace of that stimulus in the firing rate of the neurons. Now, that would be a hit. And if you present the same weak stimulus, and it just due to the stochasticity fails to reach this threshold for self-sustained activity, if you then take away the stimulus, activity collapses again, and that would be a miss. Okay? Now he modeled this, and of course we tweaked a bit, although we did not do all the effort we could have taken. And basically you reproduce all these effects that we saw in our neurophysiological data. Okay, so you get more activity on the hits than on the misses. And in the frontal cortex, of course, you have this dichotomous behavior. Whenever it goes above the threshold for self-sustained, it basically goes into this sort of high state. And you also reproduce the effects that you see on the trials without the stimulus. So you see that on the trials with a stronger stimulus that is missed, you get also this extra bump. So apparently not strong enough to kind of cause this self-sustained activity. And you see also on the trials where there was a false alarm, you get also this ramping of activity. And in the model, we know that this is due to spontaneous ignitions that happen at different time points in different trials. And if you then do the average, you get this slow ramping. And what happens here, of course, is there's no stimulus, but because of the stochasticity, it may happen that in parietal and frontal cortex, it still reaches the threshold for self-sustained activity, and then it kind of goes into the high state. Okay, so this is the modeling results. I think this was a rainy Sunday afternoon for Stan. And this is our neurophysiological data side by side. So it's a bit embarrassing, but we worked, I think, 4 or 5 years hard to collect these data. So the modelers always have a big advantage. But you see that it's quite a nice match. Of course, it's not perfect because at some point we stopped tweaking the parameters. And this is a model that is fairly simple that could account for a lot of the data. Okay, so now going back to the signal detection theory, what have we learned? Well, I would like to claim that we find that the variation in internal signal strength of the model across trials, they seem to be explained by variations in feed-forward processing of the activity towards the higher areas that can cause ignition. The threshold for perception, so this red dashed line here, is the threshold for producing this self-sustained activity. So when it enters into working memory. And maybe not so surprising, but at least in the model, we can easily explain the false alarms by these spontaneous ignitions. Okay. So much for conscious awareness of very simple stimuli. Now I'd like to go to more complex stimuli. And this is a slide that I like a lot. And here you have to do more than just detect a contour or a dot of light. You really have to, your visual system really has to work a little bit. For instance, if you want to see whether this or this is part of the same zebra, turns out that really requires a bit more time than just detection time. And what makes this task hard is that neurons at early levels of the visual system have small receptive fields. Okay, so the neurons first only seem to care about a very small fraction of the visual space, but that's not how our perception is, right? I mean, a neuron that codes for the nose and a neuron that codes for the leg somehow have to communicate to make explicit that they're responding to features of the same perceptual object. And we propose that this is solved by the brain by what we call an incremental labeling operation, where all those image elements that need to be bound into a single perceptual object are labeled with enhanced neural activity. And psychologists call this object-based attention. Okay, so if this is the stimulus that comes in, due to the interactions, also through lateral and feedback connections, the visual cortex creates this labeled representation where all those image elements that belong together are labeled with enhanced neural activity, not oscillations. Okay, so here we have the feed-forward phase, but this is then followed by the recurrent phase where feedback connections come into play to make sure that this labeling process happens in the appropriate manner. Now I want to do another psychophysical experiment with you. I'm going to present to you a stimulus, but only briefly. And your task is to see what is there. So 1, 2, 3. Did you see it?
Bird.
You saw a bird. That's amazing. Did everybody see a bird?
Nothing.
Those who did not see a bird, what did you see?
Those who saw it before.
Okay. So, I mean, this is the picture. I suppose that those who didn't see a bird did some stuff, maybe some color, brown, green. And most people, it takes some time before you see the bird. Okay, so just this very brief presentation is not sufficient for everyone. And this is, of course, where our visual system has to group together all those image elements that belong to a single perceptual object. Okay, so this perceptual grouping operation is something that I've been interested in for many years. And I worked together with Victor Lamme, who already also 25 years ago demonstrated that neurons in primary visual cortex care about a figure in the background. So here is a receptive field. Here, the image elements in the receptive field are the same as they are here, but here they're part of a figure and here they're part of a homogeneous texture. And he demonstrated that neurons, not during their first response is just determined what is inside the receptive field, but the context starts to matter at a later point in time. And after about 90 to 100 milliseconds, neurons start to have stronger responses if their receptive field is on a figure, as opposed to when their receptive field is on the background. Okay, this is this labeling process I was talking about. And here I would like to ask the very fundamental question, how does this then work in the brain? Say a few words about the role of attention. And then an important experiment that we recently completed demonstrating that these signals that we see in V1, that they might actually matter for perception. And we also recently tackled the question, does this affect and indeed depend on feedback connections or not? Okay, so let's start with a more global introduction of how this could work. And then it's always good to look at the psychological literature. So if you read the papers about figure ground segregation, the papers typically suggested that there are two processes at work here. The first process is sensitive to the features and in particular the discontinuity in features at the boundary between the figure and the background. So that would be a boundary detection process. And another process that has been proposed is called region filling or region growing. And the idea is that if you select image elements of a particular orientation, say here the left orientation, that it might be a good idea to co-select adjacent image elements with the same orientation because they're likely to belong to the same perceptual object. Okay, so you have these two processes, boundary detection and region filling. Now we would like to understand how this then is detected in the brain. And modelers have looked at this, and the typical scheme that people propose for boundary detection is iso-orientation inhibition. That means that you put inhibitory connections between neurons tuned to the same orientation. So suppose you're a neuron with your receptive field here in the background, you're tuned to the right orientation. Then given that your neighbors tuned to the right orientation are also highly active, they're going to provide you with strong inhibition. Okay, the same will happen in the center of the figure. If you're tuned to the left orientation, you get a lot of negative input from your neighbors also tuned to the same orientation, so your activity is going to be suppressed. And only at the boundary, inhibition comes from only one side. Okay, so there's sort of a relative relief from this inhibition, and you get extra activity at the boundaries. It's quite simple to understand. Now, region growing, and that's the surprise, requires actually the opposite connection scheme. It requires excited tree interactions between neurons tuned to the same orientation. So how does this work? Well, suppose you're a neuron and you're tuned to the left orientation, you respond to the figure. Suppose there was a cue there or some other reason to believe that your activity is really important. So you start to enhance your activity. And then, of course, you want to tell your neighbors, hey, we're important. So this activity, this enhanced activity should spread to the other neurons tuned to the same orientation until the boundary is reached. Okay. But now we have a bit of a problem, of course, because we want to make a neural network that does boundary detection and region growing. And these effects will cancel if you do this naively. So you have to come up with a trick. Now, the trick is to do the boundary detection, to do that in the feed-forward path, or the feed-forward flow of information, and to have region growing in the feedback pathways. Now, you can do that in a model. We work that out. So if this is the stimulus, and I'm first going to talk only about the feed-forward pathway with also orientation inhibition. So here are neurons that would respond to the figure, that tune to the left orientation. You see some extra activity at the boundaries because of this iso-orientation inhibition. Here are neurons that respond to the background, where the orientation is to the right. And here, of course, you also get the extra activity at the boundary. And if you then superimpose these two maps, you see, yes, there's extra activity at the boundary. Now, the trick is to not just do this in V1, but also in higher areas where receptive fields are larger. So if you then ascend the hierarchy, you will reach a level where the receptive field has about the same size as the figure. So here's a neuron tuned to the left orientation, and all of its neighbors also tuned to the left orientation. They're silent. So this neuron can be very active. Psychologists call it pop out. Now, if you present a larger stimulus, this area where pop out occurs will be a bit higher because receptive fields there are in a higher area, so where receptive fields are larger. Okay, so this is boundary detection and pop out at multiple spatial scales. And now we're going to implement the region filling in feedback pathways. And if you do this connection scheme in the right way, you can make this happen so that eventually also the center of the figure in V1 will be labeled with enhanced activity due to the top-down effects. Now, this has a nice aspect, this scheme, because we know from the psychophysical literature that pop-out stimuli and boundaries, that they are detected irrespective of where you're interested in the stimulus. So psychologists call it pre-attentive. And the prediction from this scheme is then that this region filling process that requires to look to the higher visual areas and back, that may then depend on attention. It may depend on the question, are you interested in the figure or not? Okay, so now I go to the next topic. What is then the role of attention in figure ground segregation? And to investigate that, a number of years ago, we did a study where we required monkeys to look at the fixation point, and they saw this stimulus on every day. But on some of the days, they had to care about the location of this figure. So this gray rectangle was not visible to the monkey. This would be the receptive field. But this square figure was visible, and the monkey was supposed to make an eye movement to the center of the figure. That was a figure detection day. But on other days, the monkeys could completely ignore the figure because they had to trace a curve that started at a fixation point to make an eye movement to the end of the curve. Okay, and this is the experiment that was done by Jasper Poort. So on figure detection days, the animal makes an eye movement to the center of the figure. And on curve tracing days, the animal can completely ignore the figure. Then we also varied the location of the figure across trials relative to the receptive field, so that on some of the trials, the receptive field would be in the center of the figure. On other trials, it would be on the edge. And on yet other trials, it would be fully on the background. Actually, I think we had a total of 23 figure locations relative to the receptive field. And this shows you the activity. So this is sort of a replication of the Lamma effect. So if you present the figure ground stimulus, you get a strong response because then now there's a texture on the screen. And you see that activity elicited by a figure here in red, by the background here in red is weaker than activity elicited by the figure being at the edge or the center of the figure. Now here we're interested in these extra spikes that are elicited by figures. So I'm going to subtract the background response in red from the figure response. And this plot is slightly complex. So the receptive field in green is smaller than the figure. So here, this is the spatial dimension. So at 4, the receptive field is in the background. At 2, it's on the first edge of the figure. At 0, it's in the center of the figure. At -2, it's at the other edge. And the -4, it's in the background again. So you see that these extra spikes are quite nicely located to where the figure is. And you see that they are elicited early on when the receptive field is on the boundary, this boundary or that boundary, and that there's a later gradual filling in of activity in the center of the figure. Okay? So this is quite compatible with our scheme. The boundaries are detected early on, and this region filling requires the loop to the higher visual areas and back, causing us a gradual enhancement of the activity in the center of the figure. Now this was on figure detection days. Now I invite you to form a mental picture what the activity should look like if the animal is not attending to the figure. Okay, do you have it in mind? This is the answer. So you get still strong activity at the boundaries, but the region filling process is much less complete. Okay, so now the animal is not interested in the figure, so you don't have such a strong feedback region filling process. Now we also recorded in area V4 where receptive fields are quite nicely matched to the size of the figure. So here we see also in the center of the figure early on extra activity. And in one study, we briefly silenced V4 activity with microstimulation. If you do microstimulation, you first excite the cells and then you have a phase of inhibition. And during this phase of inhibition, we actually saw that in V1, this activity in the center of the figure also collapsed. So that's our first evidence that this region filling indeed depends on feedback from higher areas. So let me summarize this for you. So if you're presenting a figure ground stimulus, first V1 neurons, which have a map of visual space, start to increase their activity, starting to code for the features. A little bit later, you get extra activity at the boundaries, irrespective of where you're interested in the figure or not. And then yet a little bit later, you get a filling in of the center of the figure with enhanced activity, but much less so if you're not interested in the figure because your attention is directed elsewhere, as if these non-attended figures are left in a somewhat more primordial state. Okay, so that's that much about the role of attention. Now I would like to address another very important question that we have had for many years and that was difficult to tackle. And that is, people always say to us, you have these very subtle response modulations in V1. Do they really matter for perception? Now we see this figure ground effect in monkeys. So figures elicit more activity than the background. People say it's a relatively subtle effect. So what about just silencing it? And that's what we try to do in monkeys, doing optogenetics. But we have not been very successful, I must admit. It's good to know that actually you see the same effect in humans. So we had this special patient where epileptologists wanted to see whether the epilepsy in a particular patient was caused by activity in early visual cortex. So we had one patient in which we could record spiking activity from area V3. And there we saw that also figures elicit more activity than the background. So what we saw in monkeys also generalizes very nicely to humans. But we can also do it in a mouse, and that's the good news. And also in mice, figures elicit more activity than the background. And in the mouse, it's much easier to silence activity. Okay, so we're now going to go to the mouse for a while. And we have mice reporting whether they see a stimulus on the left side of the screen or on the right side of the screen. It can be a contrast-defined stimulus or a figure ground stimulus. And if they see the stimulus on the left, they make a lick response to the left. If they see a stimulus on the right, they make a lick response to the right. And of course, you can always make sure that what is happening inside the receptive field is precisely matched, irrespective of whether there's a figure or background in the receptive field. Now the mice can do that. They find the contrast task easiest. So here they reach a level of about 90% correct. If it's an orientation defined figure or a phase defined figure, you see they're a bit worse. They're 70%. And they're not as good as monkeys or humans, but at least we have some signal here to work with. Okay, so first we're going to then do recordings. As you may know, receptive fields in mice are much larger. They're 20 degrees as opposed to, well, 1 degree or less in monkeys. So we have to also scale up the stimulus a bit. And if you then look at the activity in V1, you see that if there's a stimulus in their receptive field, then you get a response. And if there's no stimulus, you get no response. That's not a big surprise. But here's the figure ground stimulus. So here's the figure versus the background. So you see that figure ground modulation for orientation defined figures, phase defined figures, and texture defined figures. We want to know what the latency is. So the latency of the visual response is about 60 milliseconds. Of an orientation defined figure, the figure ground modulation, so the difference response here has a latency of about 95 milliseconds. And for phase and texture defined figures, it's about 30 milliseconds later. Okay, so that's nice. So we are basically reproducing all the things we saw in monkeys, also in mouse area V1. And now the important experiment that we always wanted to do, and now we can do it. So what we're going to do is we have mice, and they have, we injected the virus with an optogenetic inhibitor. It's called GTACR2. And if this is the V1 response, figure and ground, then we can knock out the entire activity from V1. It's a very efficient inhibitor. So we're really knocking out all the spikes. The nice thing is you can also postpone the inhibition a bit. So you let through the feed-forward response and you only knock out the late spikes. Or you can let through a few of these late spikes as well, and so on and so forth. Okay, so that's the manipulation. It's the onset of the optogenetic silencer. So in the contrast detection task, this is a bit of a more complex graph. I'll lead you through it. So if there is no light, so the mouse cortex is undisturbed, you see that the mouse has an accuracy here on average, what is it, 85% or so. The different symbols are the individual mice. The green symbol is the average. Now, if we silence activity early on, starting say at 20 milliseconds, you see that activity accuracy drops, but it doesn't become 50%. It doesn't become chance level. So this is some suggestion that maybe some of the information bypasses V1. It might be mediated by superior colliculus. It might be a form of mouse blindside. Who knows? Okay. And then if we then postpone the silencing to 60 milliseconds, or even better, 70 or 80 milliseconds, you see that the mouse can do the task as normal. So that suggests that for this task, they only need a feed-forward response. So V1 is basically acting at the relay. It sends the information to the higher areas, but at the later stage, V1 is not necessary anymore for the task. Now, if you present orientation defined figure ground stimuli, the profile is completely different. So you see that if we silence early on, the mice are really at chance level. So they can't do the task without V1. And we have to postpone the optogenetic silencing to, what is it, maybe 120 milliseconds or so before they can do the task as normal. Okay, that's good news. So they really seem to require this late V1 activity to do the task. And in the phase-defined stimulus, we even have to postpone the silencing by even more. Actually, that's nice because we saw that the figure ground modulation also emerged a little bit later in this task. Okay, so I think this is excellent news. To solve this task, it turns out that you need the late V1 spikes. You need the spikes in the phase where the V1 neurons express figure ground modulation. Okay, so does figure ground modulation in V1 matter for perception? I think yes, at least we demonstrated that the late spikes matter for perception. Now, a small twitch on this experiment. So we were able to address another question that we had for a long time. Does it depend on feedback? So now what we're doing is we are not injecting the optogenetic silencer in V1, but we injected it in a number of higher visual areas. As you may know, in the mouse, there is a ring of higher visual areas surrounding area V1. So this is where we injected the virus. This shows you the expression. And just an example stimulus. So again, this is when we don't silence. So figures elicit more activity than the background. This is when we silence, and we started silencing before we presented the stimulus, and we kept on silencing until we took the stimulus away. So we're basically knocking out the higher visual areas, at least some of them. And you see that the peak response is largely undisturbed. And the delayed response, or this figure ground modulation, that is to a large degree abolished. Okay, so if you look across a larger population, I think we even have more data now that there is no effect on average across recording sites in V1 on the peak response. Maybe not a big surprise because we knew that this requires input from the LGN. But the figure ground modulation is typically suppressed by, depending on the type of stimulus that we use, sometimes it's suppressed by more than 70%. So demonstrating that indeed this figure ground modulation depends on feedback from high visual areas. Okay, so I'm going to conclude this part of my talk. So I've argued that this labeling process that takes place during perceptual organization plays a role. It actually solves the binding problem. It solves the problem in locating all those image elements that belong to a single perceptual object. And it's also interesting to compare results with a very simple stimulus, where V1 seems to act as a relay and the higher areas causing ignition, and the stimulus where you need to scrutinize it for a while before you see what's going on. So there probably the features are registered, but here the higher areas need to reach back to V1 to kind of make the right perceptual organization. In some sense, you could say that the global neural workspace now actually involves the V1 neurons, right? You need the recurrent interactions with the V1 neurons to get the appropriate conscious percept in place. Okay, so I'm not sure how I'm doing regarding time. Is there still 10 minutes or so?
Yeah, we have 10 minutes.
Okay, so I would like to spend the last 10 minutes talking about how we're using this information to create a cortical prosthesis for blind people. So this will address the 40 million people in the world who are blind, and you may realize that many of them will not benefit from a solution in the retina, because in many of these patients, the connection between the eye and the brain is also lost, because the ganglion cells that are in the retina, that are the source of the fibers in the optic nerve, have also degenerated. So one possible target for a cortical prosthesis is primary visual cortex. And we already know for a long time that if you put an electrode in primary visual cortex, we are currently now using these Utah electrode arrays. And if you stimulate there electrically, then a person, could also be a person who has been blind for several years, will perceive a dot of light at that location in the outside world where you're stimulating in the retinotopic map in area V1. Now, if you place many electrodes, say 1,000 or more, you'll be able to generate 1000 phosphenes or more. We call such an artificial perceptive of light a phosphene. And the hypothesis has always been that if you then stimulate an appropriate set of electrodes, you should be able to generate a recognizable percept. You could work with it as a matrix board along the highway, right? If you flash one bulb, you're going to see a dot. If you flash the right pattern of bulbs, you can see something meaningful, a letter, a word, or some other instruction. Okay, so this is how other people envisioned that this could work. So there's a camera looking in the outside world. Then you need a processor that translates camera images into brain stimulation patterns. I hope you can recognize this as an outline of a person. That needs to be sent wirelessly to a brain chip that then activates the appropriate neurons through electrodes. Okay, that's a system that we would like to build. But one of the big questions that was not yet addressed is whether it's possible indeed to give meaningful percepts of shapes by stimulating multiple electrodes at the same time. So that's what we wanted to address. This is the work of Xing Chen in the lab, and we basically implanted 16 of these Utah electrode arrays. Each of these arrays had 64 electrodes, 8 by 8. We are not yet wireless, so this is a connector that protruded through the skin of the monkey. This was implanted on the skull, and these wires ran to the brain, and these electrodes were punched into the cortex. This shows you how we implanted the electrodes. So we tiled the visible part of V1, and we had 2 electrodes in area V4. This shows you the receptive fields of all these electrodes. So this is central vision. And this is not very surprising, but it's nice to see all these electrodes. So we can really address a large fraction of a quadrant of the visual field of this monkey. And we basically reproduced the effect also in a second monkey. And what we then did is we first mapped out the receptive fields. And I'm just illustrating to you how we did this. It's quite standard. So this is the monkey. Of course, this was not visible during the experiment, but this is just for you to see. And we're plotting the activity on top of the arrays. And you see if you then move a bar of light through the visual field, you get a wave of activity across the V1 electrodes. And you use this wave then to plot out the receptive fields. And in this movie, we plot the activity then once we have plotted out the receptive field in the outside visual world at the location of the center of the receptive field. Okay, and this is not unexpected, I suppose, but it's nice to see that a bar of light is trailed by an activity wave, and the delay between the wave and the light bar is, of course, the delay between a cell in the retina and a cell in the primary visual cortex. Okay, here we're not so interested in recording, we're interested in stimulation. So these monkeys were also trained to make an eye movement to a gray dot. And then we did not show the gray dot anymore, but we replaced it by the stimulation of one of the electrodes. Okay, because we wanted to map out the location where these animals would perceive the phosphenes. So now they're supposed to make an eye movement to an artificial perception. And this shows you that works. So these small green circles are the centers of the V1 receptive field, and the largest circle is the one that we're stimulating. And the purple dot is the gaze position of the monkey. So you see if you stimulate one electrode, the monkey is going to make an eye movement to the location of the receptive field. Okay, now this had been shown before. We are only the first to do this for hundreds of electrodes. The big question is, can we also elicit perception of shape? Okay, so we trained these animals to recognize 16 letters. And here, for instance, we present the letter D, and then the monkey gets a choice menu and is supposed to make an eye movement to the letter that he just saw. And then in the most exciting part of the experiment, we kind of replace the letter by stimulating a set of electrodes where the receptive fields would form the shape of a letter, hoping that the animal would then indeed make an eye movement to the corresponding letter. And this shows you how that works. So here we're stimulating a number of electrodes in the shape of a letter L. And you see that the monkey chooses the appropriate letter from the choice menu. Okay, and I must admit that we were really excited when we saw this result. We made a small dance in the lab. So this, I think, is a very important demonstration of proof of principle, that in principle, it's possible to elicit shape perception by stimulating multiple electrodes in primary visual cortex at the same time. Now, we're not yet there. There's still a lot of things to be developed, especially the electrode part of it. Because these Utah electrode arrays, they worked for about a year. Actually, in the first monkey, it worked maybe 11 months or so. In the second monkey, only 9 months. And then you see buildup of scar tissue around the electrodes. So this is probably not something that will give rise to a useful prosthesis. So we have to get better electrodes that have a much better longevity. And so with that, I would like to conclude. So in the beginning, I talked about how a weak stimulus reaches awareness. And then I said, it's not always that simple. Sometimes kind of getting the information to higher areas is not enough. Higher areas have to reach back. And then later, I talked about how we are now using this knowledge. I would like to mention the people who did the work. So the work on consciousness, the recordings were done by Bram van Vught, Bruno Danino, and Dev Vartak. The people working on the perceptual organization, I think there was also an important role for the people, for PhD students here on the right, that did all the difficult mouse work. And of course, Matthew Self, who has been involved in all of these studies. And the visual prosthesis work that we just published last December, was done mainly by Xing, Chen, and Feng Wang. Okay, this could be a good time to ask additional questions.
Okay, Peter, thank you very much. I want to ask everybody to unmute yourself and give a big applaud. Wonderful talk.
Yes.
And of course, I'd be happy to open the stage for questions. I want to say that there are two pending questions in the chat, but we can do them later. So
I would like to ask something. Thank you very much for the great talk. I would like to ask regarding the cortical prosthesis. Do you think that a surface electrode can do about the same work without causing any fibrosis, or maybe they can stay longer than penetrating electrodes?
Yes. So the answer is, I think they can stay longer. There is evidence for that. So there is work of Brindley, Dobell, and now recently there is the Orion prosthesis project. There has been a paper in Cell last year by Michael Beauchamp using surface electrodes. So I think the longevity of those electrodes is much more promising. The downside is that with surface electrodes, you have to give much more current. So it typically starts with 2 milliamps, and that's a factor of 100 larger than we use with intracortical microstimulation. And they find in the paper in Cell, they report that sometimes they can get 2 simultaneous phosphines. 3 is difficult, and if they go above 4, then it becomes a large undifferentiated blob. And what's going on here is they're already probably putting 10 milliamps on V1, and they're starting to indiscriminatively activating all kinds of neurons that they don't want to activate. So therefore, I think if you talk to Michael Beauchamp and Daniel Yoshor, they also start to believe that they might want to go away from surface stimulation, and that it might be better to do intracortical stimulation, because there you also get much preciser percepts and therefore a much less chance of getting interference.
Okay, thank you.
Question? I have a question. Peter, first, really an amazing and outstanding talk. I enjoyed it very much. So actually I have several questions, but I'll start just with 2. First is, so just before you published your paper, Daniel Bishop actually published another paper, I think it was in Cell, and then he used sort of like a dynamic stimulation pattern in order to generate shape perception in humans. And I was wondering, what do you think about this? Because I think that your paper actually has shown for the first time that you can actually, that the monkeys can actually perceive static contours by simultaneously activating the electrode. And I wanted to ask, do you think that dynamic stimulation can improve this, or this is just a unique example for the humans and the specific electrode that, that they were using? This is the first question. And the other question that I wanted to ask you in relation to the visual prosthesis, how do you see the long-term development of this device? Do you think that in the future, in order to get, I would say, a more natural vision, we would have to activate V1 at the columnar level, at the cell type level? Or do you think that sort of like a topographic code or retinotopic code, like the one that you were using, would be, the goal would be enough, would be the desire? Thanks.
So the first question is about the dynamic stimulation that Michael Bonchamp did in this paper. So I think that was their way out of not being able to stimulate much more than one electrode at any one time. So they basically wrote the letter N by stimulating electrodes sequentially. And that worked. So it's great that it worked. And they even had this trick that if you have two electrodes and you stimulate them at the same time, the person saw something in the middle between the electrodes. So that's how they kind of get even a more fine-grained writing of the letter N. It's going to be not ideal if you really are going to act in the outside world. Because, well, you have to suppose that you're outside and there's a car and there's a bicycle and there's a tree, and so how are you going to ride all these things, you know? And if you can only have one thing to ride with at any one time, it's going to extremely limit your bandwidth, or basically the amount of information that you can see at the same time. So therefore, I think it's much better to be able to stimulate multiple electrodes at the same time and just get the shape there right at once. So I think that's the difference. Now, the reason that this current steering, so basically getting something in the middle between the two electrodes worked, might be caused by the fact that they are stimulating in the milliamp range. So if you have two electrodes and you're both stimulating with 2 milliamps, maybe you can get something in the middle. Of course, you have to draw this current flow, and there could be one hotspot where the current is highest. With the intracortical electrodes, we're stimulating maybe 20 microamps, sometimes 50. And the insights are that what you're stimulating are the axons in the vicinity of your electrode tip. There's this nice paper by Mark Histed, who looked at this many years ago, the 2-photon imaging. And you're stimulating basically neurons that happen to have their axon there where your electrode is. And then in a sphere of course, depending on the current strength, but maybe 200 microns or so, something on that order. And in that sphere, you're not stimulating all the electrodes, not all the neurons. You're stimulating maybe only 10% or 5% of those that happen to have an axon there where your electrode is. And you cannot control the set of neurons that you stimulate. So some of these neurons might kind of code for green, others for red. So I think that's the reason why the color of the phosphine is typically whitish, yellowish. You don't have any control over that. So to really get natural vision, I don't think it's even possible with electrical microstimulation. You might need to use a sophisticated laser, right? And now using these sophisticated lasers to get single cell specific activation of neurons, that you can do it now already with maybe 10 or 20 neurons. There are some papers by Dijsselrod and I think Rafa Justa did this also. But that's not going to give rise to something that is feasible soon, because you don't want to go out in the street with a 2-photon microscope connected to your brain. It's not going to work. So for now, I think we'll have to live with phosphenes that are unnatural, but maybe if you can make them fine enough, you can still build up a relatively detailed picture of the outside world, good enough for people who are blind to gain independence, but not good enough to give you vision back as you would like to have it.
Thank you.
Thank you. Thank you, Peter. Can I ask you 2 questions, please? My name is Robin. It was very interesting talk. We really enjoyed listening to you. Very interesting advancement in the technology. So I come from biomedical engineering field. I work mostly with interbody communication. So I have 2 questions. One is related to the mice models that you have tried. A very interesting results that you have shown. My first question is, how transferable is the result in mice models to primates or humans? So what would be the motivation of doing a mice study? That's the first question. The second question is, okay, you mentioned about coming to the prosthesis part, wireless communication between a sensor, so to speak, and those electrodes. So for that, what kind of data rate would you foresee to have a realistic approach? Thank you.
So the first question was about using the mouse as a model for vision.
Yes.
And it depends on your question. So for our question, we first demonstrated the mice could perceive the difference between a figure and a background. I think that was important to establish. So apparently figure ground segregation is so fundamental that it is some process that is shared by many species, including the mouse. And that's good, right? Because then you can use all these powerful tools that you have to analyze circuits in mice that you don't have in primates. Now, other questions like whether you can impinge a pattern on the cortex and whether the mouse could recognize it, I don't think that's going to work in mice. So you always have to kind of choose your system that you want to study or choose the species that you want to study depending on the question. Also, if you want to do complicated figure grounds, perceptual organization experiments in mouse, it's not going to work because these animals just have very poor vision. And so for some of the more advanced questions, you really will have to go to the monkey. Same is true for if you want to study eye movements. Eye movements in mice are very different from eye movements in monkeys. And I could go on for an hour or so pointing out the differences between mice and monkeys. I don't think that's necessary.
Yeah, but probably you might get some fundamental understanding of the. Okay.
And, I mean, it's nice to see that you get the same thing in the mouse, in the monkey, and in the human, demonstrating that this is just the process that seems to translate very well across species.
Great, thank you. Yeah, the second question was about the data rate. So what data rate?
Yeah. So it of course depends on the number of electrodes that you want to address, and depends also on how often you want to refresh the instructions for every electrode in terms of stimulation. It depends also on the number of parameters you want to change. If you are only going to change the current amplitude, and you say, suppose you only choose maybe 64 current levels per electrode, and suppose that you only want to refresh it every 200 milliseconds or so, and if you do the math, for 1,000 electrodes, the data rate sending into the brain is only something like 4 megabit or less per second. So it's really feasible with current technology. So what I haven't shown you is that it can also be very beneficial to read brain activity while you're stimulating. So we basically found that if you activate neurons in higher areas, so we had also recording electrodes in before, that they can tell you whether your stimulus is strong enough to reach conscious awareness again. So it turns out that if you stimulate weakly in V1, you're not going to activate the V4 neurons. If you stimulate more strongly, at some point you start to activate the V4 neurons. It turns out that this moment also coincides when the thing becomes recognizable by the monkey. So in some cases, it's very beneficial to also do the recording. And recordings generally require much more bandwidth, especially if you also want to be able to detect spikes and to get the whole signal from your amplifier in the brain across. So then you need much higher bandwidth.
Excellent. Thank you. Thank you very much, Peter.
Peter, I want to read you a few questions from the chat. So there are two questions about the study that you've described before with the V1, V4 and the dorsolateral activity. So the first is, have you looked whether response strength in V1 and V4 is correlated to D prime or the criterion? And
yes, yes. And the answer is 2 times yes. It's related to, I think, a question that was asked before about spontaneous activity, ongoing activity. So we basically found that if there's a lot of ongoing activity, especially in the prefrontal cortex, that the criterion is different in the sense that the monkey is biased to say yes. And that is quite a nice fit with the model. You know, it means that you only need a little bit of extra activity before you cross the threshold. And the other question is activity related to D prime? And also there the answer is yes. If we sort the trials where the monkey saw it, actually we did something even more sophisticated. We looked at the spontaneous activity and we found a measure that allowed us to predict whether the monkey had a high sensitivity or low sensitivity. Based on those measures, then we sort of, in a cross-validated way, we chose those trials where we knew, based on the spontaneous activity, that it was likely the monkey was going to have a higher D prime. Then we saw that the activity propagated more efficiently from lower to higher areas. So further demonstrating that the kind of the unreliability of the system, the noise in the system, is related to the efficiency of feed-forward propagation of activity.
Okay, great. And that was a question by Monica Grauman, and now a question by Udi Zohari. Couldn't the attention effect be related to the planning of the saccade towards the stimulus?
Yeah, so that is from Ehud, and it's related to the task in which the monkey was either making an eye movement to the end of the curve or an eye movement to the figure ground stimulus. And I think the answer is yes. I think it's strongly related, and we know that there's a strong relationship between the planning of eye movements and shifts of attention. So that definitely played a role here, and it was even better than that. So if you have a figure, say try to outline the figure. So the task of the monkey was going to the center of the figure to make an eye movement exactly to the middle of the figure, and he did not get the reward if he was too far off the center. So the monkey really had to appreciate the entire figure to make a saccade. Now, what we found is, so in normal trials, when a monkey went to the center of the figure, I'm trying to now, so you had figure ground modulation that looked like this, like a, okay, you get it? So background, figure, 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 and then background again. Now, if the monkey was biased to the left, it was like this. If the monkey was biased to the right, it was this. So you could even predict based on the pattern of figure ground modulation where the eye would land.
Okay, that was for Udi. And last question in the chat: how the prosthesis will work on blind-born people who have never seen before? So I guess
yeah, probably not. So those people are going to learn to use that part of Cortex to do something else. So Braille, or if Ehud has even a paper on that you can even get working memory, if I'm not mistaken, for words or something like that. So very unexpected. So then if you plug in, you're not going to probably create anything meaningful for those people who have learned to use that part of Cortex differently. Now there's an interesting analogy to cochlear implants, and that's the following. Cochlear implants is a field that has been ongoing for at least 30 years. And they started, of course, to implant people who have heard, who have a normal auditory system, and then lost their hearing at a later age. But now, after all their experience and knowing that it's safe, they're now moving to babies. So they're doing cochlear implantations in babies of 9 months. Now, my prediction of this field is that those people who kind of are blind and did not develop their visual system, they're never going to be helped, right? They will have to live with their handicap. The people who have seen, so I have a normal visual system, and there we could see whether we can get this to work. It's still a big unknown. If we can get it to work, some of them might be helped. Now, if this field then develops further and we are 30 years down the line, I won't be around, I suppose. But maybe that's the time then that they will also go to babies, because babies might have, maybe with even more sophisticated stimulation techniques, they will be able to develop a system, their visual system, based on the artificial input.
Interesting. Okay, I see that there's another question in the chat. The previous one was by Inval Zelig, and this is what is the relation between region filling in you measured and the figure labeling, like the occluded zebra? Does filling in work for an attended occluded figure? If not, how does this solve the binding problem?
So it does work for complicated shapes. So zebra-like shapes, although, I mean, we're still working on that. We have seen that, for instance, for a U shape. So if you have a U shape, so with an interior, right? So then you see the labeling on the legs of the U, but not in the center of the U. So it's a very precise, very refined process that it's not like a blob of attention. It's basically object-based attention that is really kind of completely sticking to all the parts that are figural. So that solves the binding problem in a way. Now, occlusion is something else. So my prediction would be that you see the labeling on the parts that are visible. I'm not sure what's going to happen to the part that is amodally completed. So that's basically behind another object. I don't know. It could be that there are special neurons that even do something there, but I don't know. So that's something that as far as I know, people have not looked at yet, but it's an interesting question. My prediction is that you will get the extra activity for all the unoccluded parts of the object, and the occluded parts, I don't know.
Okay, and another question that popped up in the chat. How soon after becoming blind can the visual cortex change its purpose and be used for other types of processing?
Yeah, so I think this is a question about critical periods. So when is the system still able to kind of develop normally? How long can you postpone that? We know it for some animals. We know it from mice, it's I think 30 days or something like that. We know it for cats, it's I think 6 weeks or so. For humans, I mean, we should ask Oliver Braddock, I think he knows, and Jane Atkinson, they probably know better than I do. So I suppose it's maybe 4 years or so, 5 years, and then it develops further. But I think the very early years, year 1 and 2, might be even the most critical ones. But to be honest, I don't know the precise critical period in humans.
Okay. Okay, so if there are no more questions, thanks again for a wonderful and inspiring talk. It was a very pleasure having you here with us, and I hope that the recording will be okay and will go on our YouTube channel. I also want to say that next week, same time, we have Thomas Euler here, and I'll send the invitation to everybody. And again, thanks everybody for joining, and thanks Peter for joining us and for the wonderful talk. Okay,
have a nice day.
You too.
Bye.
Thank you.
Bye-bye, Sharon.
Bye.
Bye.
Bye-bye.
Bye.